In the previous video, we discussed about the neoplasm and its types. If you want to watch that video first, the link is in the description. Now here we are going to discuss about the sarcoma and its mechanism. Sarcoma is a type of cancer that develops from the cells of mesenchymal origin. In the same way, we have carcinoma, which has epithelial origin. The sarcoma that develops from mesenchymal tissue has various types from different tissues. Like we have muscle tissue, fat tissue, fibrous tissue, blood vessels, bone and cartilage, and nerves. The muscle tissue has further skeletal and smooth muscle. The sarcoma that develops from smooth muscle tissue is named as leiomyosarcoma, and the sarcoma from skeletal muscle is termed as rhabdomyosarcoma. From fat tissue it's liposarcoma. From fibrous tissue it's fibrosarcoma. From blood vessels it's angiosarcoma. From bone and cartilage it's osteosarcoma. And from nerve tissue it's neurofibrosarcoma. Now let's explore the sarcoma developmental mechanism. Sarcomas can arise through three primary genetic changes: chromosomal rearrangements, gene mutations, and copy number alterations, that's CNA. Chromosomal rearrangements. This involves structural changes in chromosomes such as translocations or inversions which can disrupt normal gene functions. Second is gene mutations. Specific mutations in genes can drive the uncontrolled growth of mesenchymal cells. Then we have CNA. These are changes in the number of gene copies, either an increase or loss, which can influence cell behavior and promote tumor formation. First of all, let's break down the key types of chromosomal rearrangements. We have duplication, deletion, inversion, and translocation. First, we see the duplication. Here a portion of chromosome is copied. As we can see in the image, the blue colored section of the gene has been duplicated. This extra copy can lead to an overproduction of certain proteins, which may promote uncontrolled cell growth. Second is deletion. In this case, a segment of chromosome is lost. The image clearly shows that a portion containing blue gene sections is missing. This loss may involve tumor suppressor genes, which normally prevent cells from dividing uncontrollably. Without these genes, the cell loses its ability to regulate growth. Third is inversions. This happens when a segment of chromosome flips and reinserts itself. As seen in the image, the blue, green, and red regions have changed their order, but remain within the same chromosome. While no genetic material is lost here, this rearrangement can disrupt the proper function of important genes, potentially leading to cancer. Fourth one is translocation. This involves the exchange of genetic material between two different chromosomes. In this image, we can see that a blue and green color segments have swapped places between two separate chromosomes. This can lead to formation of fusion genes, which may produce abnormal proteins that drive sarcoma growth. Let's continue with chromosomal rearrangement and their impact on sarcoma development. These chromosomal rearrangements can drive the formation of fusion oncoproteins, which play a major role in triggering specific types of sarcomas. First, we have Ewing sarcoma. This type arises due to fusion between EWS gene and FL1 gene. This fusion protein disrupts normal cellular function, promoting uncontrolled cell growth. Second is Synovial Sarcoma. This occurs due to fusion between SS18 gene and SSX1 gene. This fusion alters gene expression, leading to abnormal cell growth in the soft tissues around joints. Then the third one is myxoid liposarcoma. This sarcoma is caused by fusion between FUS gene and DDIT3 gene. This fusion protein disrupts fat cell development, leading to formation of tumors. Now let's move on to gene mutation part, which occurs due to oncogene activation and tumor suppressor inactivation. We see the oncogene activation drives mutations that promote uncontrolled cell growth and division. We have different kind of oncogene mutations in different pathways, like we have PI3K, AKT, mTOR pathway. This signaling pathway regulates cell survival and growth. Mutations in genes like KIT, MDM2, PIK3CA genes leads to hyperactivation of this pathway, contributing to tumor formation. Second is RAS or MAPK pathway. This pathway controls cell growth and differentiation. Mutations in oncogenes like HRAS, KRAS, and BRAF result in continuous signaling, promoting cancer cell survival. Third is hippoyaptase pathway. This pathway regulates organ size and cell contact inhibition. Mutation in YAP1 disrupts normal growth control, enhancing tumor growth. On the other hand, we have the tumor suppressor inactivation. We know tumor suppressor genes normally inhibit cell division and repair DNA damage. Their inactivation removes these protective barriers, allowing cancer to develop. First is the TP53 mutation. TP53, also known as guardian of genome, prevents damaged cells from dividing. Mutation in this gene leads to unchecked cell growth and increased genetic instability. Second is RB1 inactivation. The RB1 gene regulates cell cycle. When inactivated, it removes the brakes on cell division, promoting tumor formation. Third is CDKN2A loss. This gene encodes proteins that regulate cell cycle checkpoints. Loss of CDKN2A removes these checkpoints, allowing cells to divide uncontrollably. Together, these gene mutations, whether through activating oncogenes or inactivating tumor suppressors, disrupt the balance of cell regulation, leading to sarcoma progression. And finally, we have the CNA, that's copy number alteration, which is mediated via gene amplification and gene deletions. We see the gene amplification leads to an increased copy number of specific genes. Examples include MDM2 CDK4 in liposarcoma, MYC in various other sarcomas. Then we have gene deletions. This results in the loss of tumor suppressor genes, contributing to cancer progression. Examples include PTEN loss and NF1 loss. So this is what sarcoma is, how its developmental progression occurs. I hope you like the video. If you like it, give it a thumbs up. Do consider supporting my work on Patreon or YouTube and make sure to subscribe this channel. Thanks.